Hi everyone, this is the 2023 May paper 1R, and we're going over part 2, which is questions 5 and onwards. Alright, so question 5. A student uses this method to investigate the effect of exercise on the production of sweat. Place a person in the room which has a temperature of 20 degrees, ask the person to remain inactive for 5 minutes, then measure the amount of sweat produced by a person by a 25 cm cube area of skin. Ask the person to exercise and measure the amount of sweat produced, then let them rest. Okay, so this is done in different temperatures. And then part A1, it says, explain why the person rests for 10 minutes before the method is repeated. So this is to make sure that the person is going back to their baseline. So things like their heart rate is going back to their baseline, what their normal is. Their breathing rate is going back to their baseline. And also you want to allow for the sweat to evaporate before you continue collecting more sweat for the next temperature. So we can say this is to allow the heart rate and breathing rate to return back to normal. This is also to allow sweat to evaporate. And then part 2, it says state 3 variables that should be controlled. So examples of this would be, you want to make sure that the person is doing the same kind of exercise or the same intensity of exercise. It wouldn't be fair if one person was doing running on a treadmill or sprinting on a treadmill while the other person would be like weightlifting, for example. That might produce different amounts of sweat, right? So the same type of exercise, you want to also wear the same type of clothing, because clothing will affect the amount of heat that you feel. So if someone's wearing like a jacket, right, versus someone that's wearing a tank top, that would be different. And you also want to collect sweat from the same area of their body. So maybe like from their armpits, maybe the sweat produced from their armpits would be different from the sweat produced on their arms, for example. So you want to standardize that. Okay, let's write that down. So we can say the intensity and the type of exercise. You can say one or the other. The clothing that they wear, the area on their body in which they collect the sweat. All right, let's move on to part B. The table shows the student's results and then plot a line graph of the student's results. Okay, join the points with a straight line. All right, so this is what the graph should look like. Your x-axis is the room temperature and the y-axis is the amount of sweat collected. And then you can see that I've joined the points with straight lines. All right, part 2, explain the differences between the two sets of results. All right, let's scroll back up here first. All right, so we can see from this trend that as the room temperature increases, the rate of sweat that is produced also increases as well, but it's a lot more prominent with the people that are exercising. And that's because our muscles are contracting. They require more energy. Energy releases heat, and we need to lose that heat, right? And one of the ways in which we can cool down and lose that heat is through sweating. All right, let me write this down. So when we exercise, our muscles contract more, which means that the muscles undergo more respiration as energy is required for contraction. Respiration produces heat, which increases our body temperature to cool down our body. More heat needs to be lost from our body via sweating. As the room temperature increases, our body needs to lose more heat, which is why the sweating increases. All right, let's go over the marks. So the first mark is exercise increases muscle activity. So I said muscle contract more, and more energy is required. And there is an increase in respiration. That's the third mark. Heat is produced from respiration. That's the fourth mark, and that needs to be lost from our body. And we've got increased sweating, which is this, and then the trend, which is as the room temperature increases, the rate of sweating increases. All right, let's move on to question 6. The lining of the inside of the cheek is made out of squamous epithelial cells. Draw a labeled diagram to show a cheek cell when seen under a light microscope. So it's asking you to draw a cheek cell, so we're just going to draw something that looks like this. And then we're just going to draw the nucleus in the center. So this is the cell membrane. You need to label that. You need to label the nucleus, so that is the nucleus. And then lastly, the cytoplasm. All right, part B, describe how a sample of cheek cells would be prepared to view using a light microscope. So for this, we would need to use a cotton bud to swab the inside of the cheek to get the cheek cells. And then afterwards, we would transfer them onto a microscope slide. Then we add a stain and then add a cover slip on top. Okay, so to obtain cheek cells, first swab the inside of the cheeks, then transfer it onto a microscope slide. Then add a stain, followed by covering it with a cover slip. All right, let's go over the marks. So we've got to obtain, you need to first swab the inside of the cheeks. That's the first mark. Then transfer it on a microscope slide. And the third mark is you can either say add a stain or cover it with a cover slip. But both these points are important to do. All right, let's move on to part 3. State 3 differences between a cheek cell and a cell from the lining of the trachea. All right, so on the left we have some cheek cells. On the right we have the cells that line the trachea. One difference is that with the cheek cell, they're more irregularly shaped, whereas the cells in the trachea, you can kind of see them here, they're more rectangular, or they're more oblong. Right, the second difference is that the nucleus of the cheek cells are located in the center of the cell, whereas here you can see that the nucleus is located near the bottom of the cell, right on one side. The third difference is the cilia. So on the cheek cells, you don't have any cilia that's present, but here in the trachea, you can see that there is cilia, and the purpose of cilia is to brush away the mucus. So the first difference is the cheek. The shape of the cheek cell is irregular, whereas the cells in the trachea are more oblong in shape. All right, the second point is the position of the nucleus in the cheek cell is at the center, whereas the cells in the trachea are the nucleus is near the bottom. And lastly, there is cilia present in the trachea cells, but hold on, let me just correct that. Trachea cells, but not in the cheek cells. All right, so those are your points. Okay, let's move on to question 7. The 4 blood groups that occur in humans are group A, B, group AB, and O. These are the 3 blood group alleles, and IO is recessive to both IA and IB, and their IA and IB are both codominant to each other. Explain what is meant by the term codominant. Okay, so we can say when the alleles are codominant, it means that when they are both present, they are both equally expressed. All right, the next part, state the possible genotypes of a person with blood group B. So there are 2 possibilities here, either IB, so homozygous dominant, or IB, IO, which is heterozygous. We know that this allele is recessive. All right, let's move on to part B. Haemophilia is a sex-linked condition that prevents the blood clotting. Explain why males are more likely to be affected by haemophilia than females. All right, so this is how we can write out the answer. Haemophilia is caused by a recessive allele on the X chromosome. Females have 2 X chromosome, whereas males only have 1 X chromosome. Because of this, females would need to inherit 2 faulty alleles for them to have haemophilia. If they are heterozygous, they are carriers and they do not have haemophilia. All right, so I'm just going to go over the marks and then also go over some other points you can also say. So the first mark is it's carried on the X chromosome. Whoops, why is it not coming out? Okay, X chromosome. The second mark is that it's not expressed in heterozygous females. The third mark is females have 2 X chromosome, and another mark is males only have 1 X chromosome. Other things that you can also say is only females can be carriers. And another thing you can also say is males can't be carriers of that disease. All right, let's move on to question 8. The diagram shows a bacteria cell. Name the parts labeled A, B, and C. All right, let's start with let's start with letter A, which is the flagellum. So the flagellum helps them to swim and to move around, right? B is the cell wall. So the purpose of the cell wall is to prevent the bacteria from bursting. And C, that is the loop of DNA, right? So we have the loop of DNA, or you can just say DNA. You can also say genetic material. All right, part 2. Some types of bacteria can cause disease. Give the name of organisms that cause disease. That term is called pathogen. Pathogen. All right, let's move on to part B. A student uses this method to investigate whether antibiotic X is more effective than antibiotic Y. So first, they grow the bacteria on a nutrient agar in a petri dish, which is what we see in this diagram here. And then here they'd soaked this filter paper in antibiotic X, and then this one they'd soaked the filter paper in antibiotic Y. And they place it into the petri dish, they seal it and leave it for a few days, right? So basically how this works is that the larger the diameter, so you can see the larger the clear zone, you can see the clear zone here as well. So the larger the clear zone, the more effective that antibiotic is at killing the bacteria. All right, let's start off with part 1. Describe what is meant by the term antibiotic. So this is specifically what the syllabus would want you to say. An antibiotic is a substance typically produced by microorganisms to kill bacteria. So 1 mark for produced by microorganisms, and the second mark is to kill bacteria. All right, let's move on to part 2. Explain which antibiotic, X or Y, is more effective against the bacteria in the Petri dish. All right, let's scroll back up to the diagram. So earlier I mentioned that the larger this clear zone is, the more effective the bacteria is. And in this case, the clear zone for antibiotic X is much larger than the clear zone for antibiotic Y. So X is the better antibiotic. All right, let's write that down. Okay, so antibiotic X is more effective than Y because it has a larger clear zone. This is because when the antibiotic diffused from the filter paper to the nutrient agar, it killed a larger number of bacteria. Okay, part 3. Explain a suitable control that could be used for this investigation. So you could dip the filter paper in probably distilled water. And this is a great way to see that if there is any clear zone, it's because of the antibiotic and not because of the filter paper, for example. The filter paper can be soaked in distilled water only without antibiotics, as it does not produce a clear zone. This is to ensure that any clear zones produced is due to the effect of the antibiotic. Okay, let's go over the marks. So the first mark would be filter paper soaked without any antibiotics. The second mark is there should be no clear zone, or you can say that should not affect the bacterial growth. And then the last statement, which is this is to ensure that, that's supposed to be any clear zones produced is due to the effect of the antibiotic. All right, so that is the end of the 2023 May paper 1R. I'll be doing the paper 2 later on soon. If you have any questions, just leave them in the comment section below, and check out more videos if you're interested. All right, bye.